Good afternoon, everyone. It's certainly a pleasure to be here on behalf of Twist to talk about some of the early exome data that we were able to look at, particularly in some FFPE samples using the kit, and again, a very recent data set. These were done really within the last month or so. So it's an early look at the data, but overall, our impressions have been so far favorable, and I'll certainly describe that to you in the next couple of slides. I oversee the clinical and research production laboratories at HudsonAlpha, and one of those in the Genomic Services Lab, as you see on this chart, it does a lot of projects from a lot of different locations over the world, and we can make all kinds of goofy graphics like the odometer you see there, which is roughly the number of times Chris Mason's name appears in the program this week, or equivalent to the number of bases that we sequence. And for those, I know Chris isn't here, so I can joke with him, and he'll get me back tonight at our session. So older samples in FFPE, whole genome and whole exome sequencing. This is a slide that's probably a year, maybe 2 years old. And I just want to highlight some of the numbers that are shown in green, particularly duplication percentages, which are shown on your far right, the total sequence, which is the column next to it. Notice it's hovering around 10 gigabases of sequence per sample, and then the coverage metrics hovering in the low 90% for 30x, numbers rising as you get to the 1x, 4x, 8x, et cetera. And then particular attention to the last two rows, so 27 and 25% duplication rates, 10.9%, and hovering around 94%. So this was about as good as we could get with a highly optimized exome prep that we had been doing previously on a wide diversity of FFPE samples. And in this case, low input meant right around 100 nanograms or so material. No other quantitation other than just pico-green quantitation. So nothing to do with amplifiable fraction or anything more specialized than that. So keep those numbers in mind as we go forward a bit. We've had an opportunity to work with a large number of very diverse samples. This represents actually a snapshot of about 9 months of isolations and processing in 2017. The peripheral blood being the largest source of samples, of course, because this is generally matched germline and tumor samples, blood being the most common germline match. And then in the neighborhood of a few hundred to several hundred samples, depending on sources. So many of these are from both, or these samples come from both retrospective as well as prospective studies. So that means there's quite a range of age and quality of the fixed formalin paraffin embedded tissue. But again, it's a very important, I'm sure as everyone recognizes, very important tissue source for biomedical research. So our evaluation criteria in this small test we did was as follows. Input requirement, lab workflow, flexibility, and then performance across the diversity of samples were the overall goals. And the workflow is summarized in the slide you see here. So in this case, 50 nanograms of input material was used for each sample in a 10 microliter volume. And importantly, the preparation methods, the fragmentation, end repair, and add A all occurred in one tube, one step. No intervening purifications and no other intervening steps other than adding the appropriate reagents, incubation, followed by, and then each of the blue arrows indicates a purification step. So a purification into ligation, then into PCR, followed by purification, and then a quality control step. That quality control step essentially was just quantitation. And then in terms of the standard protocol, it was 8 samples per hybridization. Each sample was added in as 187.5 nanograms of final library after low cycle PCR. Hybridization takes place, to emphasize, in a pooled basis. So there's washing, PCR, and then final quality control, which was quantitation again, insert size determination, and then of course real-time qPCR to determine the molar sequenceable fraction of that final pool. So the experimental design was we made 2 pools. So again, a relatively small experiment, 16 samples total. So in pool number 1, it had one replicate each of 2 HapMap samples, NA12878 as well as NA12911, and then 6 germline samples. So high quality DNA represented by the samples you see over on the left-hand side of the gel image. Pool 2 was a replicate of 4 of those samples, the 2 HapMap controls as well as 2 germline, and then 4 independent FFPE samples. So 2 key points to mention is this means in pool number 2, we did indeed pool FFPE samples, low quality FFPE samples with high quality genomic DNA. It was done intentionally, and we wanted to see what the relative performance was in the pooled hybridizations with this diversity of sample inputs. The lower gel shows a kind of an average FFPE sample quality that we've been seeing across these studies. And then the sequencing was done on the NovaSeq. Each pool was run in 1 lane of an S2 flow cell. And so this was done with the XP workflow. And note that the yield, probably not surprisingly, and for anyone in the back that isn't able to see it, you can see the overall base quality shown in the green block was about 93% of bases at Q30 or above. And so exceptionally good sequencing run in this case. The total yield off of that flow cell was just over a terabase. So that's vastly over Illumina specification, just as an FYI. But overall, a nice sequencing run, and one that we were pretty encouraged by the overall performance of the data. So the indexing, two important things from this slide. In this particular case, the indexing was done with a standard dual index read, shown at the top. 4 different I5 indexes, 4 different I7 indexes used in a traditional dual indexing, where the example shown below it is each of the I5 indexes was pooled with each of the I7 indexes, giving a diversity of 16. And I'll mention a little bit later in the talk about some of the considerations for this sort of indexing design when it comes to sequencers like the NovaSeq. But in the case of this experiment, what this means is we didn't have an opportunity to quantitate nor control for index hopping. And I'll mention a little bit of that in the future, I'll mention that a little bit in the future of the talk. Again, this is an early version of the kit, and I'm quite confident you'll see revisions to the indexing schemes in the short term. Over on the left-hand side of the slides, though, notice, so remember, pool 1 is all high-quality genomic DNA samples. And then pool 2 is the last 4 columns represent the 4 FFPE samples. The first 4 columns are the 4 germline samples. So not particularly surprising that we saw a small bias in the sequencing against the FFPE samples, but remember, this is also taking into consideration the 8-plex capture. So with coming in with equal amounts of material into library prep, equal amounts into hybridization, and no particular quality control for the overall quality of the sample or otherwise, it's actually a pretty amazingly balanced pool set, both pre and post capture, all the way through sequencing. So again, very encouraging that it looks like it should be very possible. Again, this is an N of 4 low quality samples with 4 high quality samples, so take care not to extrapolate too far. But early indications are that mixing a diversity of sample types within the same pooling scheme and within the same hybridization scheme may work very, very well. So coverage looks strong, and since this was on the NovaSeq, obviously it's 8 samples per lane, high yield on the sequencer. It really ended up with about a 500x coverage exome, so these were essentially meant we were right around 500 million reads per sample at a paired 100. So again, quite a bit of data, an exceptional amount of data for an exome. And recall we talked about a 20% duplicate rate on 10 gigabases of data. And in this case, where we have essentially about nearly 10 times that amount of data for some of the samples, the duplication rates were still hovering in the low 20% range with 50 nanograms of input material. So clearly we were seeing a highly complex library that captured quite efficiently. And so we're actually, frankly, very surprised at the complexity of the library and the ability to sequence with maintaining a very reasonable duplication rate percentage all the way out to 500x coverage. Capture efficiency, really about where it was expected, which is shown in the columns on the right-hand side, which is both percent in capture strict and percent in capture plus bait. And then, of course, the 20 and 30x numbers. And it probably comes as no surprise at 500x coverage, all of the metrics at 20 and 30x are well over 99%. The last thing to point out on the FFPE samples is note the discordant rate, which is shown on the 5th column on the side. For anyone who can't read the numbers, it's higher on the FFPE samples. What I think is happening is a combination of both the lower quality sample as well as we know in these particular sample types, with this particular index scheme, the index hops are higher. When the index hops are higher, the discordant rate goes up. And so that's a factor in this as well. But the overall capture was very strong, remained strong in the FFPE samples, actually a little bit better than some of the genomic DNA, and that's very likely because the average insert size on the FFPE samples was slightly smaller, so it likely captured slightly more efficiently. And it's given that the library preparation methods, enzymatic fragmentation plus the other steps were equivalent between the two, there may be some opportunities for slightly optimizing that in the future. So obviously a 500x exome is extraordinarily deep coverage and really isn't leveraging the decreased cost per sequence that the NovaSeq allows. And since we didn't have an opportunity to empirically or directly run lower coverage, what we did is we downsampled that same data set and downsampled it to 4 gig, 6 gig, 9 gig, and 15 gig of data that you see across the top, giving both mean and median coverage. And again, as Ramsey mentioned, if you look at the reasonable correlation or fairly tight metrics between mean and median, it again speaks to the uniformity of the capture. So we're quite pleased to see that. And also that what it means is 9 gig of data is really what would be for about a 100x exome. 4 gig of data give you around 50x or so, and then 6 gig, of course, 68x. And the FFPE samples fell well in line with the germline controls. So this chart, I'll walk through everybody through it, and then I'll explain why I made sort of this not really complicated, but sort of bizarre looking chart. Each of these bars delineates the replicates. So this is the 4 control samples. This chart is all the FFPE samples, and this chart is the 8 germline samples, which is why so this is 4 gig, 6 gig, 9 gig, and 15 gig, and then what you're looking at is Percent of base is at 150x coverage, 100x coverage, 30x coverage, 20x coverage, and this is 4x and 8x, which in all of the plots are essentially flat at nearly 100%. So that's the 99% and above. And you can see as the amount of data steps up from 4 to 6 to 9 to 15, you can look for the step, largely a step function, or at least on average. And again, this is each of the replicates, where you can see here this particular replicate fell out from the others. In this case here, the germline samples were a little more consistent. And this step function, as you go from 6 gig to 9 gig, 9 gig to 15 gig, if it's fairly linear and proceeds in this fashion, it actually helps speak to the complexity of the library, which is why we generate the plots in this format for this kind of downsampled data. And so in this case, the FFPE samples, again, a bit more challenging. So you see these plots move around a little bit depending on the sample. That's not unexpected. But overall, as you get out to the 15 gig, this is where you really see the 100x coverage being up above 90%. In this case, the 100x coverage up above 90%. And in this case here, this is the 100x coverage up above, and then this included 150x coverage in this particular plot. So overall, you can decide, depending on where you want saturating coverage at what percentage. If 20x is your number, this is the 20x line up here in green. So very, very quickly, even at 6 gigabytes of data, gigabases, gee whiz, of data, you can get there pretty fast. So the next question, of course, is concordance, right? How well do the replicates have concordance in the exome capture? Not surprisingly, they're also quite high. This would be actually a fairly expected result, I think, so greater than 99%. Notice the variant, the total variance and number of counts. So this is the full coverage, the full 500x. So we're actually looking at concordance across about 150,000 variants in each case. And then same thing for the concordance on the downsampled replicates, total variance and concordant variance. Notice they rise as the data rises, and the reason simply for that is that the variance that made it into this calculation had to have a coverage of at least 20x with a PL score of 30 or higher. So high quality variance with reasonable coverage went into the calculation, and not surprisingly, as you increased exome coverage, you increased the number of variants that went into that calculation. But note that the concordance rates still all remain pretty strong. Now, some of you may be wondering, why isn't it 99.9% or so? And a lot of these are intronic variants. Certainly, there's some spurious results because of alignment issues in some of those regions of the genome. So if you instead restrict it to a concordance between, where we look at only overlapping bed file regions of two different capture products, one being NimbleGen, the roast NimbleGen product, and this one being the Twist product, you again now see those 3 nines of concordance across a smaller space of variants. Again, looking at only the intervening regions, or essentially the overlapping protein coding regions of the exomes here, and then we see those concordance rates jump back up as expected. So overall, the results were actually very encouraging. Again, FFPE samples showing also equal concordance among them, equal variant counts, indicating that they were performing extremely well in this pilot project. So the future experimental design, I think what this means overall is you could imagine something like this, that you have a 96-well plate shown here. Those 96 samples become 12 hybridizations in an 8-plex. Those 12 hybridizations then becomes 2 different pools of 6 hybridizations per pool. You do that 2 times with 192 samples and put that on an S4 flow cell, 4 pools per flow cell using an XP workflow. Or if there's enough indexes available, again, you could keep them uniquely indexed. And at the bottom line, that should give you right around 100x coverage per exome, or 10 gig of sequence data at a paired 100 sequencing condition. Now, some of you who are familiar with the S4 flow cells say, well, but Illumina only sells that in a paired N300 kit. That's fine. We found really no advantages to running out past paired N100 for the exomes, and so we actually just throw those reagents away. The increased value per read on the S4 more than makes up for that difference. A very reasonable experimental workflow, in a typical laboratory design for probably mid and high scale work overall. So as far as what does that mean for exomes on the NovaSeq? So 16 samples on an S2 gets you about 40 gigabases of data, or that'll be right around that 500x coverage. That's what was done in this study. If you increase that to 48, 96, 144, or 192 samples, you can kind of see how this scales: 140x, 70x, 45, and 30. And then same thing, obviously, on the S4, it's about 2 times the output, so you see more output. And then what does it come down to dollars and cents? Is probably these would be the 2 most common mechanisms I would predict that people would use. And this is just sequencing reagent costs at Illumina list price. So it's really not sequenced cost. It just gives you a comparator between the 2 conditions. Probably about under $100 in sequencing reagent costs for a 45 exome, and you're probably under $200 in sequencing costs on a 100x exome. Again, respectively on the NovaSeq. I did want to mention, first, we found the workflow to be highly efficient, very convenient hybridization protocol in terms of its quantitation into a pooled hyb. really an exceptional balance over a range of sample qualities based on things we've seen in the past, and quite low duplicate rates under very high coverage conditions, which has also been an area, at least in FFPE samples, that we found some struggles. And then, of course, the expected strong concordance that you'd predict from a product that's accurately capturing the exome region. But so indexing is an important issue, and I did want to mention that on the NovaSeq, and this is something that Illumina has published application notes on, and I think is becoming reasonably well known, that oversimplifying indexing is a bad idea, and here's some data to show why. This picture here is from a app note that Illumina put together. It's available on their website that talks about the different chimeric molecules that can happen in an index hopping event, and that's just some examples here. In the interest of time, I won't go through those other than to say having libraries that have dual unique indexes, meaning a unique I5 and a unique I7 in the same molecule, which means you need 192 different indexes to make a 96 index set that would be required, is the ideal index set. And moving away from any reuse of a given index in an experiment is actually critically important. But it's not as simple as just coming up with 96 indexes that work in a certain way. As you can see here, if you look at these just kind of a little bit arbitrarily numbered plates, and notice this overrepresentation of some of these indexes, and this is some testing on a dual unique set of 96 indexes that Angela Jones in the lab has done. And notice there's some periodicity here that you can see, and that pattern is real, and that pattern repeats on a plate basis that has nothing to do with the samples that are run on them. So it's entirely related to the index combinations. Taking the exact same indexes and just reordering them, you can get to a fairly flat pooling here. So it's not only the index content that's important, but actually the combination of indexes. Stated another way, very, very simply, it means I'm suggesting that all of our data shows that the difference of 16 nucleotides over the course of the entire library insert actually makes a difference in how it sequences in a reproducible manner. So that's why I say oversimplifying an index scheme is actually has consequences, and indeed needs to be looked at quite carefully when you consider designs like I suggested on the earlier slides of 192 samples across an S4 workflow. Because in the most convenient form is you'd make one pool and avoid an XP workflow. But in this case, hopefully I can show you that doing dual unique indexes can be optimized and can be highly reproducible. And I think I can be confident in saying that it's highly likely you'll see these types of designs coming out from companies like Twist. So final word of acknowledgement of a pretty amazing laboratory group, in particular Angela Jones, who really did all of this work in terms of both the running of the protocol. So any detailed questions on the sequencing, feel free to grab her, and then of course both clinical and lab personnel. And then the other thing that it was struck by for some Emily's early comments, I think it's really come full circle when we talk about now sequencing whole genomes and now talking about taking data from our whole genome sequence and then storing it in DNA, right, as a storage solution. So, right, I mean it's just right. Anyways. So anyways, thanks for your attention, and I'll happy to take some questions after the session if anybody has any. Thanks.